ATP. Here's the molecule right here, and we'll talk about what it is and how it's used. So, adenosine triphosphate is the name, ATP for short. This is essentially used as a storage of free energy. And remember, when we say free energy, we're talking about the ability to do work. So this molecule essentially stores energy so that we can get work done. There used to be these things called traveler checks, which you could take around to any different place and you would be able to use them as money there. They're kind of like that, in that if you think of the money's the ability to do something, ability to do work, these are our cellular version of that. They are produced and then used throughout the cell to do all sorts of different work that needs to be done within the cell. ATP is the high form. When you have a high energy form of it, you have all three phosphates on there and it has the ability to do work. And then ADP, you've gone from triphosphate down to diphosphate. This has broken off one of the phosphate groups, and that's the uncharged version of it. It has to be regenerated into ATP for it to do work. When it does that work, it gets turned, it loses that phosphate and it becomes ADP, and then it needs to be regenerated. So here's the structure here, and a couple things to note is that, as tends to be the case in biology, we do a terrible job of tracking hydrogens. And so here's a version of it without the hydrogens on the phosphates here. You may see versions where they actually have these oxygens bonded to hydrogens, and the idea is that that is what they can do, but since this is biology, we're going to continue the tradition of not doing a great job of tracking hydrogens. When you take your ATP and turn it into ADP, you are losing one of those, not that one, because that's the second one. We're losing one of those phosphate groups. It becomes unattached and it becomes a free phosphate ion. But by doing that process, you have the ability to do work. Here's a more colorful version of the same process. Again, you can do a hydrolysis to get that phosphate off there, and the OH can be attached on there, and again, it's going to be a little bit more complicated in this. Often it's not normally just getting attached to a hydrogen itself, but it may be attached to something in the middle before it becomes unattached to it. But essentially, this removal of a phosphate can be used to get work done. One thing to note is that it often looks like by breaking this bond, you would get energy out of breaking the bond, but of course that's not true, right? Because it always takes energy to break bonds. So if you were to break this phosphate off here, that would require an input in energy to get it to be broken, but once you attach it to something, you'll get an output in energy. And as long as you have a greater output in energy, the reaction will be spontaneous. So it is not breaking off the phosphate that gives you energy, because that would actually require energy. It is the coupling of that with the formation of some other bond, depending on the situation, that is where you get your free energy from. As new bonds are formed in the products, that's where you're getting your energy back out, more stability. So this process of taking that phosphate group from the ATP and changing the ATP into ADP, this phosphate group will end up stuck onto something, and that attachment of it, that rebonding, the reformation of bonds, that's where you get the energy out of it. It's a coupled process, and the net outcome, if it's going to be spontaneous, is going to be that it is releasing energy. So again, another picture of it here, but this is focusing on the regeneration of ATP. So realize that this is a cycle. You're going to be taking your ATP with your 3 phosphates, and we're going to remove one of those phosphates. And again, it's not just breaking the bond, it gets attached to something, and then there's some more to the process here. But the net result would be a drop in free energy, so it'd be a spontaneous reaction overall. But anyways, then this ADP has to be turned back into ATP, so we will have to regenerate that. And note that often we don't want to draw the whole inorganic phosphate ion. And if we do have the inorganic phosphate ion, it's actually got one of the hydrogens attached to it, but often we don't have that hydrogen attached, drawn, or we don't track those hydrogens, and then often we just write a P with a circle around it. So just a P with a circle around it, that is the phosphate ion that we're talking about removing from our ADP. Summary, we don't really keep a good track of those hydrogens. So for example, let's say you wanted, for whatever reason, to take this reactant, glutamic acid, and ammonia, and you wanted to add them together. Lots of fun, you would get glutamine. But the problem is, if we look at the free energy for this reaction, it ends up being a positive value, which you should remember, we won't get into the calculation for it, but it is a positive value, meaning that it is going to take up free energy. It is a non-spontaneous reaction. It requires work. All those referring to the same thing. Positive delta G requires work, non-spontaneous. So this reaction won't happen on its own. So what we do is we say, okay, well, let's break it up into steps. And as long as each of the steps are spontaneous, we'll be able to carry out the reaction. So what we can do is we can take our glutamic acid and use ATP to phosphorylate it. And it turns out that that has a negative delta G, a negative free energy, which means it's a spontaneous reaction. And what we can then do is take our glutamyl phosphate, is what the intermediate reactant is called. We can take this phosphorylated version of glutamic acid and then add it to the ammonia. So we're sort of broken up into two steps. That second step is also has a negative delta G, which means it's spontaneous. So you've broken up this process into two steps. Each one of them is spontaneous, and therefore you can have the reaction happen spontaneously. And think about this, right? There is no free lunch, right? This is not the exact same process as above. So this first process and the second process, the difference between them, of course, is you're using ATP. So you have to destroy an ATP molecule in order to get this process to happen. That's what ATP is for. This is why we use it as a form of storing energy. The process without it won't happen on its own. It is non-spontaneous. But if you use ATP in this particular way, break the reaction up into two spontaneous steps, by destroying ATP, you can then have a spontaneous reaction. So two spontaneous reactions will add up. And again, remember that the breaking of these bonds is going to take energy, but by forming the bonds, so breaking up the phosphate and then attaching it to this glutamic acid to make glutamyl phosphate, that overall ends up being a spontaneous reaction. Here it is in a more graphical sense. Again, this particular process of attaching the glutamic acid to ammonia is not spontaneous. You've got to put work in. It's got a positive delta G. We refer to that as an endergonic process, also non-spontaneous process, positive delta G, all the same thing. But we know that if you take ATP and you remove the phosphate group from it, that is a spontaneous reaction. That will happen on its own. It is an exergonic process. So if you were to tie these guys together, then what you can do is as you have this exergonic process happening, you can use it to do an endergonic process, and then you can get what you want done spontaneously. So we couple these reactions. Metabolism is all about coupling an exergonic reaction with an endergonic reaction in order to have them happen. Net is a net free energy, net delta G is a negative value, and that means it is spontaneous. So endergonic reactions require an input of energy. Exergonic reactions release energy. What we do is we tie those two processes together. That's metabolism. And this allows us to have the processes we want done to occur. So cells couple the exergonic hydrolysis of ATP with some, and again, this is the great thing with ATP, is it can be used to do so many different things, some endergonic reaction. We tie them together, we couple them together, and this allows the endergonic process that we want to have happen to occur. This ATP has a change in energy. So here's the enthalpy of negative 30.5 kilojoules per mole. But again, that it is an exergonic change when it lets go of that phosphate. We can use this exergonic process. And if you've done some chemistry, you may refer to it as you say, okay, well, that's a delta H and it's negative, so it's exothermic, which is true. But also, what we were factoring in when we talk about the energy, the free energy, not just the heat energy, but the free energy, the ability to work, we refer to that as exergonic, and that incorporates the idea of entropy in as well. So not just heat energy, but also entropy and the temperature at which it's occurring, the whole Gibbs free energy equation, all of that gets calculated in, and that term we use then is exergonic. So we can use this exergonic energy, this free energy, to put into bonds. We can actually build larger molecules and store that energy in bonds itself as chemical energy. We can use the energy for mechanical energy, things like moving the cilia, the flagella on cells, contracting muscle fibers. All of that is going to require some energy. We can get that from ATP. And we saw with transport, we can move particles from a low concentration to a high concentration against what would happen normally through diffusion. We can fight diffusion by expending energy by using ATP. Heat can also be used, so the loss of energy is always going to be happening through all of these processes, so we can actually use the processes themselves to generate heat. It's not normally intentional, though we do have mechanisms to purposely generate heat when we don't have enough of it for us endoderms. So, free energy applied to sugar. If you take a sugar, you can burn just a bunch of sugar, and it will give you carbon dioxide and water with a huge decrease in energy. You can't do that in a cell because it'll kill the cell trying to give off that much energy at once. So what metabolism does is it still does the same process. We still have the overall expansion of energy, but it's broken up into many, many tiny steps, each one manageable by the cell, giving you all of that free energy in the process, but in a very controlled manner, one little step at a time. Redox reactions, again involving electrons, so redox reactions involve a change in electrons, is another way to store energy. We're going to see this with two electron carriers throughout the process of metabolism that we're looking at, cellular respiration, and again in photosynthesis. So these electron carriers can pick up electrons, and kind of like an ADP can pick up a phosphate to form ATP, and that is the higher energy. It's storing free energy like that. These electron carriers, NADH and FADH2, can pick up electrons, and when they're holding onto those electrons, they are the high energy version. They have the ability to do work. So here is the diagram of it, and there's our empty electron carrier. It can pick up the electrons, in which we call it NADH. That's the full version. NAD plus is the empty version, and NADH has more free energy than NAD plus. So we're essentially adding in free energy, and now if you have the NADH, you have the ability to do work. We can couple that with some process we want done, and we can get work done. Same thing happens FAD and FADH2. FAD is the lower energy version, FADH2 is the high energy version. So once you add in those electrons, you are now at a higher energy version. You have the ability to do work, which can be coupled with a endergonic process in order to get it done. Notice our neglect of the hydrogens. NAD plus will pick up electrons and become NADH. This is the high energy version. It can then lose those electrons and become NAD plus again, letting go of those electrons. So electron carriers carry free energy when they have those electrons, and take a look at our neglect of hydrogen.